60 individual posters. I think that's remarkable, especially given that most of the poster, but today the old-fashioned in-person, so welcome everybody here for being in person, but then also welcoming everybody online. So making it all work together took us a little while, and I apologize to that, especially for our speaker. I would like to acknowledge the support that we get for the poster session and the food and beverages that will be supplied by the Carl Zeiss Microscopy Incorporated, Google, the University Credit Union, and VWR International. So please, the next time you Google something, Google for Zeiss and VWR and the University Credit. Introduce Dr. Boyden.
Pleasure to introduce our speaker today. Dr. Ed Boyden is currently at MIT, but has a really interesting sort of career history where he started as an undergrad there studying electrical engineering and computer science and physics, so sounds like a double major there, and then made this interesting shift to neuroscience throughout his career, you know, crossing these interdisciplinary boundaries that can be very challenging to do. So he went to Stanford as a postdoc, working with but graduate student first, right? It's a graduate student at Stanford working with Jennifer Raymond as her first graduate student, and Richard Sien, and then postdocing there as well with Carl Zeiss Roth and Mark over there as a Helen Hay Whitney postdoc. Multiple awards. I mean, there's just so many, I can't list them all, but, you know, pretty much every award that you've heard of, I think, you know, he's received here, including, you know, NSF Career, NARSAD, Sloan, NIH Directors Innovator Awards, and others. And then, you know, at MIT, he's currently the Y. Vatan Professor in Neurotechnology and an investigator at the Howard Hughes Medical Institute, and is a tenured professor in the Department of Brain, Cognitive Science, Media Arts Science, and Biological Engineering. So what I really love about his, you know, career, the Center of Neurobiological Engineering, Neurotechnologies, to not only study the brain, analyze it, but, you know, have this translational focus and effect to ultimately repair the brain. And through lots of collaborations, you know, we discussed having been co-advised during his career by multiple, you know, advisors in different fields, but also serving as a co-advisor to a lot of students, where these students can really, you know, combine multiple expertise and tools across labs, across departments. So, you know, it's a really privilege to introduce him, especially as the director of this Neurotechnology T32 program that we have here at Translational Neurotechnology. And thank you, Dr. Boyden, for meeting with our trainees this morning. It was very exciting for them to see many of them who have similar backgrounds in, you know, moving across disciplines. So with that, thank you again, and over to you to give our talk today.
Thank you so much. It's an honor to get to talk to you today, and I'm sorry I can't be there in person. My employer had a bit of a travel restriction, and by the time it had been released, my teaching schedule filled up, and I wasn't able to come out this week. To talk to you today and to share a bit of what we're. So I direct a group at MIT that works on technologies that enable the mapping and as Nanthia described, I started my training as an engineer and then benefited from some great biological training. And this whole co-advisorship strategy, our group is very fond of. So at this point, almost half of our graduate students have co-advisors where, you know, build and apply technologies to solve those problems, or the inverse, some much better chemist or much better device engineer or much better molecular engineer, and then we can look like biologists. So it's kind of fun being in the middle and thinking about how the problems and the solutions collide. So I'm going to share my screen now. Is the screen sharing starting to work? Let me see.
Yes, Dr. Boyden.
All right, thanks. So our group has a focus on the brain, but as you'll see, I think that the kinds of problems we face in neuroscience are actually felt by many other fields in biology. It's just that they are glaringly obvious when we are dealing with something as complex as the brain. And the brain, I think, really exemplifies that many biological systems have vastly differing spatial and temporal scales. So let's think about spatial scales. We have molecules, cells, brain circuits, entire brains. I mean, we're dealing with orders and orders of magnitude difference in spatial scale. And then temporal scales also are daunting. The briefest events in the brain last a millisecond or even less in duration, but a lot of the great processes we care about, whether it's learning and memory, or the progression of Alzheimer's disease, or other one thing that really motivates me is to the point of making neuroscience into, well, you know, a very mature science where we can make predictions, we can make computational models, we can make hypotheses that can be tested in a very systematic way, the way that mature sciences like physics and chemistry have been doing for a century. And I think our field, of course, is very much an art form as much as it is a science. So we developed tools that really try to confront three kinds of questions. How can we make maps of the brain? How can we look at and control the high-speed signals of the brain in a way that's easy to do? We believe in making tools that anybody can use, but that really help get our community to confronting these sort of ground truth-oriented, systematic questions about the brain. Now, as I sort of alluded to when I began the seminar, the brain really presents these enormously challenging spatial and temporal differences. And so for each of these challenges, whether it's mapping, controlling, or watching the processes of the brain, we've had to really take the spatial and temporal scales into stride. So for mapping the molecules throughout brain cells and the brain cells throughout neural circuits, the first half of my lecture, I'll talk about a tool suite that we've been building that I call expansion microscopy, where we are enabling anybody, we hope, to do nanoimaging across extended three-dimensional spaces. And again, we had to really confront the extended spatial scales of the brain head on. In the last half of the talk, I want to tell you a couple short stories about the control and observation of high-speed signals in the brain. And again, the vastly differing temporal scales over which brain circuits operate present unique challenges. So I sometimes only half jokingly tell people in our group, our challenge is to help biologists conquer space and time. We want to help people map and understand and control biological systems across all these different scales. So let's jump right in. The first half of the talk is about space. How can you understand the 3D configuration of molecules throughout cells and cells throughout tissues and organ systems, and even sometimes entire organisms? Brain cells are enormous, right? And the human brain, they can be many centimeters in spatial extent. The ones that go down our spinal cord project a meter approximately in length. But axons and dendrites, synaptic connections, the molecules along those axons and dendrites and at those connections, those are all nanoscale and often organized with nanoscale precision. So many people have developed techniques that let you look at nanoscale things. Electron microscopy, super resolution microscopy, these pioneering tools have helped people look at very tiny objects, even individual biomolecules. But they all struggle to deal with large 3D objects like brain circuits, and they often require equipment that can be costly or require specialized training. So in our group, we often start projects by wondering, what if we do the opposite of what everybody's doing? For literally 300 years, people have used some kind of lensing strategy to magnify images of objects in biology. What if we made biological systems literally larger? And so we call this strategy expansion microscopy. The basic concept is, can we chemically create a dense spiderweb-like mesh of swallowable polymer, kind of like the stuff in baby diapers, throughout a biological specimen? So these polymer threads shown in the cartoon on the left will wind their way inside cells and outside cells, around biomolecules and in between biomolecules. If we anchor biomolecules or labels to that polymer, soften up the specimen a bit with chemical treatment, and add water to swell the swallowable polymer, maybe we could pull all the building blocks of life apart from each other until they are far enough apart that we can resolve them with an ordinary microscope. No need for any specialized equipment. You can just use your ordinary hardware to do nano imaging. So this idea owes a debt to two old lines of research. I never got to meet him. I guess he unfortunately passed away relatively young of a heart attack. But an MIT professor, Teuchi Tanaka, was working out in the late 1970s and early 1980s the physics of swallowable polymers. And he has this paper that's been cited a couple thousand times where he looks at swallowable polymers where when they absorb water, you get this volumetric change that's very fast and very large. And in the early 1980s, their team worked out a bunch of the physics of how this rapid, huge change occurs. They actually studied sodium polyacrylate, the active ingredient in baby diapers, and the actual material that we use to do our expansion process. And the physics that they worked out suggested to us that maybe there's something here that we could apply to evenly or isotropically expand brain circuitry. Now, the other thing you have to solve is you have to get this swallowable polymer inside the brain, right? You can't just dump it on top. Amazingly, around the same time, in 1981, Christine Dreyer and Peter Hausen had this wonderful paper where they used free radical polymerization to create a dense spiderweb-like mesh of polyacrylamide, a very similar polymer, but uncharged, throughout preserved embryos and tadpoles, actually, amphibians and so forth. They created hydrogels throughout these preserved specimens and showed, 1981, that they could use these hydrogel-embedded specimens to facilitate staining and imaging. So it's fun to think about, if somebody had connected the dots between these two areas almost a half century ago, maybe the talk that I'm giving you now could have been given then. Okay, so what do we want to do? What we want to do is to take a cell like the one on the left, this golden neuron, and turn it to something like the cell on the right, a constellation of biomolecules hovering in space. Their 3D organization preserved. Two biomolecules that are touching will be pulled to some minimum distance apart from each other, and molecules that are some distance apart from each other will be scaled up by a linear factor. So you end up with this constellation of biomolecules hovering in space, but pulled apart by the swelling of the polymer. Now, to make this work, we have to invent a number of chemistries, which are illustrated in the following cartoons. In this cartoon, proteins are shown in brown. We had to develop anchors, handles, that could bind to biomolecules and help us pull them apart from each other. So here in purple are the anchors binding to the protein shown in brown. And since our original paper, we now have developed anchors that can bind to RNA, DNA, and proteins, and hopefully soon, working with groups like Laura Kiesling's and others, we're hopefully going to have ways to look at sugars, lipids, and other important classes of biological molecules. Next, we have to create that spiderweb-like mesh of swallowable polymer. And we use a strategy that's a lot like the 1981 and 1980 papers that I just mentioned. We use free radical polymerization to create a mesh of sodium polyacrylate. The monomers are shown in white spheres, and they self-assemble into chains of polymers, shown as white lines. When a polymer chain grows and encounters a handle, shown in purple, it forms a covalent bond. So if you think about it, the polymers, shown as white lines, can swell apart from each other. The handles, shown in purple, apply the force to the biomolecules. We're almost there, right? But there's one last thing we had to develop, a way to soften the specimen. Because the specimen doesn't like being expanded, it's very happy where it is. And so we have developed chemical, heat, enzymatic treatments that allow us to soften the specimen, loosening up things. We add water, and now the polymer will swell. But this time, because of the handles, all the biomolecules or labels will be pulled apart. So we first announced that we could do this in 2015 to then graduate students, Fei Chen and Paul Tilburg, who now both direct their own groups, made this work. And panel B is a piece of mouse brain tissue. And panel C is the same piece of mouse brain tissue. About a day, day and a half later, we've polymerized, softened, and expanded it by about 100 times in volume, about 4.5 times in each direction. And the polymer, I'll use the mouse pointer here, starts out quite dense. The mesh size, which is the technical term for the spacing between polymer threads, is very tiny, just 1 or 2 nanometers, smaller than many biomolecules. And after we expand, the polymer has extended its conformation. So the biggest question that we always got in the early days is, well, is the expansion even? By design, the softening, the dense nature of the polymer, by design, we had the protocol intended to be even. But of course, this is biology. We have to prove it. And so in the early days, we spent a lot of time taking pre-expansion images with a classical nanoimaging method, like a super resolution method. And then we would take post-expansion images with an ordinary confocal microscope. And here are some sample images shown in purple and green, respectively. We calculated the distortion and found out it's really small. It's not 0, to be upfront, but it's a few percent over a typical microscope's field of view. And that turns out to be important enough for most biological questions of our time. We can also look at the resolution by examining known structures like microtubules or other structures that have been well characterized with classical nanoimaging methods. Here are some microtubules in a cultured hex cell decorated with fluorescent antibodies. We can take what we see and then deconvolve it by the known ground truth, thanks to decades of nanoimaging by earlier methods, and we can estimate the resolution. Now, with a 4.5-fold expansion, you might guess that a 300 nanometer resolution lens would now have a resolution of 300 divided by 4.5, or around 70 nanometers. And indeed, that's what we find. Now, you might think, well, if the mesh size or polymer spacing is only 1 or 2 nanometers, if we expand more, can we get better resolution? And as I'll show you later, that's true. But even with 4.5-fold expansion, there are many things we can do that are difficult, if not impossible, to do with earlier technologies. I'll just give you an example here. On the left-hand side, we're zooming in from top to bottom on a piece of the mouse brain. This is a Thy1 YFP mouse expressing yellow fluorescent protein, shown in green, in some neurons. And then in magenta and blue, the color code is shown on the left, are post and presynaptic proteins, Homer1A and bassoon, decorated with antibodies bearing fluorophores. And so each white square is a zoom in of the panel above. Now, the left hand, we're zooming into a piece of the mouse brain. You can see the cortex and the hippocampus. That's before you expand, and very quickly you hit the resolution limit of your microscope. This is a confocal microscope at our local core facility. The right-hand side is the same specimen, same fields of view, but after we expand. And now we can cleanly resolve dense compartments like synapses. In fact, if you look in the lower right, the purplish blobs on the left can now be cleanly resolved into pre- and postsynaptic protein densities. And if we measure the distance between those protein densities, we get the same answer that Catherine Dulac and Xiaowei Zhang measured using storm microscopy, a super resolution method invented by Xiaowei. But now we can take these images on a regular confocal microscope. Collaborating with Eric Betzig, another super resolution pioneer, Ray Gao, Sho Asano, and Gokula Padula, we looked at different compartments of neurons, such as mitochondria and lysosomes, shown in the upper right, myelination in the lower part of the slide, again, all visualized with fluorescent antibodies. And with expansion, we could easily start to resolve these very fine, even nanoscale compartments of neurons. By the way, if you expand something 100 times in volume, it becomes about 99% water. And so it becomes completely clear. And this is what I think got Eric Betzig interested in using one of their light sheet microscopes to scan our expanded samples. So you can do expansion and do nanoimaging on a regular microscope. But if you have a light sheet microscope, you can then go at blazingly fast speeds. And so in this paper that we published about 2.5 years ago, we showed that we could go almost 1,000 times faster than an equal resolution classical super resolution method. So far, I've been showing you natural proteins decorated by antibodies, but you can also express engineered proteins, such as fluorescent proteins of different colors. We can deliver the genes that encode for fluorescent proteins into the mouse hippocampus, shown at the bottom here, and we can look at the wiring of the brain. In the top middle is a zoom in to the white square below, where we express fluorescent proteins and then image these brain specimens on a confocal microscope. And so it's hard to see these very fine wiring of the brain. But in the upper right is the same field of view as the upper middle, but after expansion. And now we can resolve individual axons and dendrites. And so many groups are now using some kind of combination of color coding plus expansion to look at synapses, axons, and dendrites in brain tissue. So far, everything I've showed you has been protein visualization, but you can also visualize RNA and other biomolecules. When Oswasi joined our group, we started working on RNA visualization. Don't just anchor the proteins, but also anchor RNA and expand them away from each other, and then do post-expansion in situ hybridization to visualize individual RNA molecules. On this slide, you can see many examples of pre- and post-expansion images of the same cell, and you can see after you expand, much better resolution than before you expand. And this can be done, of course, with intact brain tissue. Here, we're expanding again the Thy1 YFP mouse, anchoring both proteins and RNA. Green shows the YFP, and my purple dots are post-expansion in situ hybridization. And so now you can visualize individual RNAs with nanoscale precision in intact brain tissue. Now, many groups have been pioneering multiplex ways of looking at many RNAs at once. And so we started wondering, if we expand and then do multiplex RNA imaging, could you look at dozens or even hundreds of expressed genes? In work that was spearheaded by 5 people in our group, Shahar Lawn, Dan Goodwin, Anis Sinha, Oswasi, and Fei Chen, we, collaborating with Aviv Regev, George Church, and others, sought to adapt in situ sequencing into expanded brain tissue. In other words, expand the brain, bring in a hybridization probe bearing a barcode, and then sequence the barcode. Now, when you sequence something, you're just copying it. And as you copy it, you're blinking out its genetic code because the bases of nucleotide that you're adding are fluorescent. And so here, we decided to look at dozens or even hundreds of messenger RNAs, bring in barcode-bearing probes and bind to the RNAs, and then sequence those barcodes using standard chemistries, like the kind you would find with any standard DNA sequencer. Working with the Allen Institute, who's been doing a lot of RNA sequencing in the past several years, we looked at the cell types of the mouse visual cortex. We did in situ sequencing of expanded visual cortex, going after a couple dozen different genes that they had identified reflecting cell types in the visual cortex. We clustered the resulting cells, shown in the upper left, and the upper right are the very cells, but now you can see the color codes determined by the clustering in the upper left superimposed over the brain itself. So layer 1 of the visual cortex is on the right, layer 6 is on the left, and the different colors reflect the different cell types in the visual cortex of the mouse brain. Of course, with expansion, you can get resolution that is difficult to achieve with other multiplexing strategies. And so we can expand and then do this multiplex analysis even within nanoscale compartments like synapses or dendritic spines. So inspired by work from Aaron Schumann and others, we decided to look for dendritically enriched RNAs. And so we took some data sets acquired by her group and other groups and synthesized these kinds of barcoded probes against a couple dozen different dendritically localized RNAs. We expanded a piece of mouse hippocampus and then brought in those probes and then sequenced the barcodes. And what we found was we could visualize with nanoscale precision the location of dozens of different RNAs in dendrites and synaptic compartments. That's shown in the lower left. The gray is the neuromorphology. These are YFP expressing neurons. And then the red dots with the green text indicate the location and identity of different RNAs. And we can find patterns. So we can look at the distribution of RNAs in apical versus basal dendrites, or apical versus basal spines, or even look at different cell types and look for patterns in how RNAs are distributed in neurons. And it was very exciting to see that maybe there are some interesting patterns that might be more general, determining how RNAs are trafficked throughout brain cells. As a side note, this doesn't involve expansion, but opportunistically, we collaborated with a couple groups, including Jason Boden-Wasser's group at Harvard and Fei Chen's group. This is work done by Andrew Payne, Zach Chang, and Paul Reginato, and showed that we could do something similar for DNA as well. Bring barcodes into the genome, sequence the barcodes, as well as their adjacent genomic DNA, and we could do in situ genome sequencing, looking at the sequence, but also the structure of the genome inside cells. So this does not involve expansion yet, but we're hoping to bring expansion into the mix so we could do mapping of nanoscale compartments of the genome inside cells, such as in the brain. Okay, back to expansion. How big can you expand? Well, everything I've shown you so far is expanded by about 4.5 fold. When JP Chang was in the group, we worked out an idea, which was, could you take a specimen of brain, polymerize and expand it? That's the blue polymer in the cartoon at the top. Can we form a second polymer, shown in brown, in the space opened up by the first expansion and expand it again? It's like exponential spatial amplification with a linear number of steps. Kind of like PCR, but for space. Well, recently, we just had this paper accepted at Nature Biomedical Engineering. Dublina Sarkar, Jinyan Kang, and Oswasi, with great help from Margaret Schroeder and Christina Kitko and Ibuchi Emanari, have developed important versions of the strategy. We came up with a strategy that does double expansion, but using all off-the-shelf chemicals. Form a polymer, anchor the proteins to that polymer, expand, form a second polymer, pull the proteins further apart, expand again, and you can then label them with antibodies. And the cartoon at the top highlights an important byproduct of this process. You can pull proteins apart and enable better labeling. So in the top part of the cartoon, you can see that the red and green antibodies can bind to proteins on the outside of the synaptic protein cluster. But once you pull the proteins apart, shown in the bottom half of the cartoon, now the yellow and blue antibodies can also label their targets. We've decrowded proteins from each other and made room for antibodies to bind. We wondered, could you apply this to dense protein complexes like those in synapses and reveal more information? Well, the answer is yes. So we did a lot of pre-expansion staining shown in yellow, and there's not much yellow to be seen. And then same antibody, same specimen, we did post-expansion labeling shown in purple. And you can see a lot more purple. So in panel B is a calcium channel subunit. Panel C is RIM12, a presynaptic protein. Panel D is PSE95, a postsynaptic protein. In all three cases, we get a lot more purple staining than yellow. We get better labeling of proteins after we expand them away from each other than before. Now, not all proteins are crowded. In panels F, G, and H, we saw similar labeling between the yellow and the purple, the pre and the post. We wondered, though, if some of these proteins, by being decrowded from each other, can be better labeled, would it help us see interesting 3D features? So we collaborated with Tom Blanfield's group at U Maryland and wondered, now that we can see these calcium channel subunits, can we see if they're coordinated with other synaptic proteins? And this is sort of a crowded slide, but the take-home message is yes. We can see coordination of the presynaptic calcium channel subunits with other pre and postsynaptic proteins. They're not uniformly or randomly distributed. They're coordinated in what Tom likes to call nanocolumns between the pre and postsynaptic sides of synapses. This could be very important for understanding the nanoarchitecture of how synapses signal efficiently. Collaborating with Li Wei Sai at MIT, we wondered, what about very dense protein aggregates important for neuropathology? And one of the things that we were interested in investigating were amyloid plaques in Alzheimer's disease. There's a lot of back and forth about what these amyloid plaques really do or mean. And so we wondered, regardless of one's hypothesis, wouldn't it be great to see them better? So looking at 5X FAD mice, which have amyloid plaques, a mouse model of Alzheimer's, we did pre-expansion staining, shown in yellow, and post-expansion staining, shown in purple, with antibodies against A beta 42. And I hope what you can see here is that there's a lot more purple than yellow. In fact, there are even these little amyloid dots, which we did not see pre-expansion at all, and which might represent particularly dense aggregates of amyloid that are so dense that they're difficult to stain before expansion. We saw this with multiple different A beta antibodies, and we never saw them in wild type mice. So right now we're exploring very actively what these different aggregates might mean, what is inside of them, what's around them, and hoping to map out a wide variety of very dense protein environments in brain cells and beyond. So as I mentioned at the beginning of the talk, a lot of the chemistries that we have been using are really basically combining the 1980 and 1981 papers that I mentioned. But we started wondering, polymer chemistry is an active field. Can we do better? When Ray Gao and Jay Yu were in my group, we wondered, what about more even polymers? Now, the free radical synthesized polymers that we've been using in expansion have defects, shown in the upper left. Areas that are denser, dangling ends of the polymer, loops. Now, polymer chemists have invented a lot of chemicals and polymers based on those chemicals that are more even in their 3D configuration. And the cartoon on the right illustrates one of these so-called diamond lattice polymers that, as you might guess from the name, form these almost crystalline-like structures, although they're not actually crystals. And we started wondering, could those be used to expand more evenly? Well, now we're getting down to very, very fine scales. And so we wondered, how do we get down to the fundamental building blocks of life? Can we have some other way of expanding a target that lets us gauge the target's accuracy? And so we thought, let's use viruses. So collaborating with Rachel Neve, we started expanding HSV1, herpes simplex virus. What we found, shown in the upper left, was that this diamond lattice hydrogel, or tetragel as we call it for short, gave much more evenly expanded viruses than the free radical synthesized hydrogels, which is abbreviated PAAG here, shown in panel C in the upper left. In the lower right, in panel G, you can see the population data. The classical hydrogel gives us probably 15 to 20 nanometers of error in the expansion process. Not bad. But the tetragel gives us about 9 nanometers of error. So this is very exciting to us. What if we could get resolution of expansion down to that of single biomolecules? And maybe relevant to our time, we did find that the expansion of the virus with this diamond lattice hydrogel was able to be much more precise than with the free radical hydrogel in terms of preserving shape. You can apply these strategies to human tissues. Dongshin Zhao and Octavia Bucur, when they were working with me and other groups like Astrid Wines and Andy Beck and others, we showed that we could apply expansion to human tissues. These are normal on the left and cancer-containing on the right, biopsies from human patients. And we've now looked at prostate, lung, breast, pancreas, and about a dozen different human tissue types. And expansion can be used on all of them. So let's summarize the first half of the talk. We've discovered that we can physically magnify biological specimens. You can use it to visualize proteins, RNAs, and other kinds of biomolecules. You can apply this to a wide variety of tissue types from a wide variety of species. So this slide isn't really meant to be read, but I think we hit a milestone recently. The 300th experimental paper or preprint doing expansion recently came out. These are not review papers or commentary papers, but actual experimental papers. It's being used to look at how the flu virus spreads from cell to cell. It's being used to look at the fruit fly brain. It's being used to look at the human kidney, the microbiome, and the list goes on and on and on. So clearly there's demand for a way of doing nanoimaging that doesn't require expensive or difficult to use hardware. And All of our expansion protocols are on our website, expansionmicroscopy.org. We used to have lots of hands-on workshops and to have visitors come by our group nearly every week. Of course, that's been difficult with COVID. So during the peak of COVID lockdowns, we posted on our website a step-by-step photographic tutorial of how to do expansion, including very basic skills like how do you handle a cover slip, how do you build a little gel box out of cover slips and slides, how do you use paintbrushes to handle gels, and so forth. And our hope is that this is really going to help democratize nano imaging. So please give it a try if you're interested. Don't hesitate to get in touch with me if there are any problems. We're always eager to hear feedback of any kind. And maybe now it's time to transition to the second part of the talk. Obviously, the limitation of expansion microscopy is that it does not work on living things. You can only expand preserved, meaning non-living, specimens. And so in the last part of the talk, I would like to give you a couple of short stories about the control and imaging of living things. Let's start with optogenetics, although I don't want to spend a lot of time on it because most of the tools that are in use are tools that we published or that our colleagues and collaborators have published over a decade ago, frankly. In optogenetics, we use light to control neural activity. This started when Carl Dijsselroth and I actually were both students at Stanford and just started brainstorming how are all the forms of energy that we could deliver to the brain enumerated. And there's only so many kinds of energy that you can deliver to the brain. There's mechanical force, there's electromagnetic fields, there's light, which is a specific kind of electromagnetic energy. And we thought light would be really great if you could use it. You could bring light into the brain with an optical fiber or other technology. The next question became, how do we get the neurons to sense light? Because most of them, of course, do not. And so I became really fascinated by microbial opsins. These are 7 transmembrane proteins, like the one shown in the lower left. They sit in membranes of single-celled microbes that grow in salty water or in the ocean. There's a little image of that in the upper left. And they respond to light and convert light energy into chemical energy. The first to be found was a light-driven proton pump named bacteriorhodopsin by its discoverers. It has a vitamin A relative called all-trans-retinal inside the protein. That's the actual light sensor. Light hits it, causes the all-trans-retinal to isomerize, and you pump an ion, in this case a proton, across the membrane. A primitive form of photosynthesis, if you will, storing light energy in chemical form. That was found in 1971. About a decade later, several groups found light-driven chloride pumps from the same actually set of single-cell microbes. They respond to light by pumping chloride ions across membranes. And then, around the turn of the century, several groups working together found light-driven ion channels. Being ion channels, they don't convert light into stored energy, they dissipate energy, but they're used by single-celled algae and other critters to sense light to help them navigate around in bodies of water. So flash forward to the current day. What we and our group members and our colleagues and collaborators have found is that all of these classes of molecule contain members that are safe enough, fast enough, and powerful enough to be used in brain cells, even in awake behaving animals. So when Brian Chow and Xue Han worked with me, they showed that there are light-driven proton pumps that you can put into brain cells using standard gene delivery methods. Shine yellow or green light on the brain cells, they pump protons out, and you can shut down brain activity. And so these molecules have become quite popular for silencing brain activity. Xue and then later Amy Chuang worked on light-driven chloride pumps that can cause similar effects, although they differ in their biophysical details. Amy in particular showed that she could find light-driven chloride pumps that respond very well to red light. Red light is convenient because red light can go deeper in the brain or the body than other colors of visible light. And then finally, light-driven ion channels contain many members that can be put into brain cells to let us control them as well. So the first of these that we put into brain cells was done with myself and Carl Dijsselroth working together. We showed that we could put channelrhodopsin 2 into brain cells and shine blue light on them and activate them. Nathan Klepecki later found a red light activated molecule, crimson, and then Orshamesh, Yonker Cho, and others have been working to extend the toolbox to their physical limits. And so these tools are very widespread. We can deliver the genes for these molecules using viruses or transgenic methods into brain cells. We disseminate all of them freely throughout the neuroscience and biological communities. And literally thousands of researchers are using these tools to activate and shut down brain cell activity. By activating brain cells, you can figure out what their activity can initiate or sustain in terms of behavior or pathology or the remedy of pathology. And by shutting down brain cells, you can figure out what they're needed for in terms of the same kinds of questions: behavior, pathology, and the remedy of pathology. An interesting development that happened recently was when crimson, which is the red light drivable molecule that Nathan found when he was a student in my group, was actually shown to be used in humans. So a European team showed that they could take the crimson molecule, put it into the eye of a blind patient with retinitis pigmentosa, and then wearing a pair of goggles to project light onto the retina, this person could have partial restoration of vision in an otherwise untreatable condition. So it's very exciting to think about this field of optogenetics, which is widespread tool use in basic science, is now also making the translational leap to humans. But what strikes me so much about this is just how lucky we were. In optogenetics, we really engaged serendipity more than anything else. We found that there were molecules that were safe enough, fast enough, and strong enough from the natural world that can convert light into meaningful electrical activity of brain cells. And we've all benefited from that serendipity. And that's a great segue to the final set of short stories that I want to tell you about, which is that the opposite, watching signals in neurons, has not been so easy. The natural world evolved these molecules that we use in optogenetics, but the natural world has not evolved molecules that all by themselves will let us observe any neural signal with light. So when Erica Jung and Kirill Pyakovich were postdocs in my group, we wondered, if the natural world won't evolve these molecules, why don't we do it in the lab? Directed evolution, of course, has yielded a lot of very powerful tools for biology, and Francis Arnold and others even won a Nobel Prize for their work developing this field. But it still has been difficult to generate neural tools because they have to be really great, right? Like for optogenetics, you need safe molecules that are also fast and strong. They have to be multidimensionally good. So we wondered, how do you find multidimensionally good molecules? And we thought, let's build a robotic microscope. Take a gene and make millions of mutants. For any given goal, some are better and some are worse. Put them into cultured human cells, and then with a microscope, you could image those cells and figure out which cells have the gene products that do what you want. They're fast, they're safe, they're strong, whatever your criteria are. Bring in a robotic arm and suck out the cells, and therefore the mutants that are better, and there you go. You have your better genes. And this robot that we use is a commercially available robot that anybody can use. And so we did what might have been at the time, maybe it still is, but at least at the time it was, I think, the largest mammalian cell-based directed evolution screen to make proteins better. We started with a fluorescent voltage indicator candidate, Quasar 2 from Adam Cohen's group at Harvard. The reason is we wanted to start by developing what you might call the opposite of optogenetics, a fluorescent voltage indicator which will light up or glow when a neuron is active. We did 2 rounds of directed evolution, almost 10 million mutants of Quasar 2. And we found a molecule. Here's some work we did with Shahan's group at BU, where we expressed the resulting molecule, Archon, along with a somatic localization tag. We call the resulting molecule SOMARCHON. And what you can see here is that even with a very simple microscope shown in the upper left, is that we can record the voltage of neurons in an awake behaving mouse. So what you see in the middle of the slide are what look like electrode traces, but they're being recorded by a microscope on a camera. We shine a red laser onto the brain and collect the red and infrared light that fluoresces on a camera, and we can image the voltage activity of neurons. Being a microscope method, you can image populations of neurons. So here are about a dozen neurons in the hippocampus, shown on the left, and during a period of time, 8 of them, shown on the right, exhibited electrical activity traces. We think this worked well because we optimized for 3 different parameters. We wanted molecules that were well localized to the membrane, a proxy for safety, that were strong, in this case they're bright, and had good temporal properties. In this case, the molecule quasar 2 was already quite fast, so we selected for good lack of photobleaching. But I think this really highlights the power of multidimensional selection. And now I think we're seeing many preprints on bioRxiv and others who are thinking about multidimensional selection in mammalian cells with our extended network of collaborators. And it's very exciting to see the potential here for developing a wide range of genetically encoded tools. To be upfront, we were not the first to, of course, try to make genetically encoded tools for imaging. This is a screenshot from Jin Zhang's database from her lab at UCSD. Literally hundreds of genetically encoded fluorescent biosensors have been made. Basically, you take a fluorescent protein, like the green fluorescent protein from jellyfish, you fuse it to a sensor domain, and now you can, if you do it right, have a fluorescent sensor of some signal, like calcium or protein kinase A. But we were starting to wonder in our group, wouldn't it be great if you could look at many signals at once in the same living cell? After all, cells take in many inputs, they generate many outputs, and inside is this roiling sea of activity within the cell as it computes. Now, usually what you might do if you're an imager is to make indicators of different colors. Have one be green and another one be red. And now you can see 2 different signals with 2 different colors. What if you want to do 3 or 4 or more? Or what if the signal that you want to see has no sensor of the right color? Well, Shannon Johnson and Che-Yang Ling-Gu in my group, we started working on a very different way of thinking about this problem. What if you could put different sensors at different points in a cell? So the points labeled 1 might contain fluorescent reporters of calcium, let's say. The points labeled 2 might contain fluorescent reporters of protein kinase A, for example. I'm just going to take a sip of water here. While the cell is alive, each dot of fluorescent indicator will blink out a little movie of its signal activity history. So the points labeled 1 would tell you what's going on in terms of calcium at those points. The points labeled 2 would tell you what's happening with protein kinase A. After you're done, you can figure out which signal came from each puncta by staining and labeling in a preserved tissue using techniques like those I described in the first half of the talk. You can figure out which signal came from each puncta. We found a couple ways to make this work, the best of which we think is to take different fluorescent reporters and to connect them to different self-assembling peptides. They will assemble safely, randomly, and enduringly at different puncta in the cell, and each dot will then report a different signal. Each reporter also has an epitope, which you can then stain for with an antibody to figure out after the fact which signal came from each point. Here's an artist rendition made by the Ella Maru studio showing the concept. The live cell has a lot of green dots in it. You can image a little movie from each dot. When you're done, you stain, which yields the false color image and tells you which signal came from each dot. Does it work? Well, in B is an image of a cultured neuron with the much loved calcium indicator GCAP6F. Panel C shows GCAP6F expressed in a cultured neuron, but this time fused to self-assembling peptides that form these stable clusters. We did tons of control experiments to look at the amplitude of the signal to noise, the kinetics, the safety of the signal, the safety of the reporter, the quality of the signal, the nature of the cell's endogenous physiology. The list goes on and on and on. And so we don't think these clustered reporters change the cell health or the physiology. But what they do allow is that you can measure many signals at once in a living cell. Here in the upper right, you can see a green image. We expressed in a cultured neuron 3 indicators made by other groups: GCAM6, the calcium indicator, CAMP-BAR, the cyclic AMP indicator, and X-ray ACAR, the protein kinase A indicator. Panels B and C are zoom-ins to the neuron, showing in green the live cell image, and the false color image is what you get with antibody staining against epitopes that distinguish the 3 indicators. So you can take the live movie of the green cell, then do the false color imaging after you're done, and figure out which signal came from each green dot. And what you can now do is look at many signals at once in the same cell. So here we are looking at calcium, cyclic AMP, and pKa when we give this cultured neuron forskolin, a drug that drives cyclic AMP production. And now we can see the relationship between these signals. So for example, some neurons shown in H had slow calcium responses to forskolin. Some shown in I had fast calcium responses. We wondered, calcium is sort of an input to this network. Can you predict the protein kinase A output from the input? Well, as shown in L, you can. So the blue trace is the protein kinase A output in the slow calcium neurons. It's a weak output. In red is the protein kinase A output. It's a strong output. You can express these sensors in the living mouse brain. We then cut an acute brain slice and did imaging of it with a confocal microscope and found similar relationships. In panel D, some neurons shown in red had fast calcium, some shown in blue had slow calcium, and in E, the ones that had fast calcium had strong protein kinase A, and the ones that had slow calcium had weak protein kinase A activity. So we were able to use this strategy to look at the relationship between the speed of one signal and the amplitude of another signal in the same cell. We can express 4 different indicators. Here we added a protein kinase C indicator, and we can now look at 4 different signals at once. We can add a 5th indicator. Here's an ERK kinase indicator, and we can now look at 5 different signals at once. So in summary, we're hoping that we can help look at many different signals at once in a living cell. So to end the last part of the talk, in contrast to optogenetics, where we're just lucky, we now have seen that we can be a bit clever through evolution in the case of making better voltage indicators, or through strategy in the case of trying to use existing indicators in new ways, like the clustering, and help image hopefully more kinds of signal and more kinds of signal in the same cell. So to conclude, we're very excited now that there's a lot of different tools that we can build that help people map systems, control them, and observe them. In our own group, we're very excited to apply this to 2 sets of questions. One is, can we make detailed maps and data sets that allow us to model small brains like those of fish and worms? When Limor Freifeld was a postdoc in our group, she showed that she could apply expansion microscopy to the larval zebrafish, which has about 100,000 neurons. J.U. did something similar for C. elegans with 302 neurons. So we're very excited about integrating these tools to look at small brains. We also, through many collaborations, also have a focus on trying to understand the aging brain. Can we use these mapping and imaging and control tools to understand what makes the aged brain more prone to conditions like Alzheimer's disease? I think I've acknowledged along the way all the people who've led these projects, but I want to end on this slide because there's an even longer list of people who have helped at the top in our group and our alumni. And then in the middle of the slide, an even longer list of collaborators all over the world who we've worked with. Please visit our website, synthneuro.org, if you want to learn how to use these tools. And don't hesitate to email me. Just my name, Ed Boyden at MIT.edu, if we can help in any way. And with that, I'd be happy to take questions.